So welcome everyone. It's lovely to see so many of you here. Just as a bit of kind of housekeeping, we have to keep your cameras off and not contributing on chat just because we've had the odd terrible Zoom bombing experience, but hopefully you can all see each other and who's logged on, and any questions, pop in the chat for me and I will read them out later. So that's how this will run this afternoon or morning, depending on where you are. Just to introduce myself, I'm Georgia Mason. I'm director of the Campbell Centre for the Study of Animal Welfare, and we're at the University of Guelph, which is on the traditional territories and treaty lands of the Mississaugas of the Credit. And it's my great pleasure today to introduce Professor Brian Key. Brian is at the University of Queensland, and he's speaking to us from there today. We're at 7:30 in the morning, for which many thanks, Brian. And it's Thursday. It's also spring, so really quite different from here. And I was thrilled to have Brian contribute to this series because I really admire his rigor, his logic, his ability to cut through the crap. And he's really produced some very critical analyses of work to date purporting to show that fish are sentient. I don't always agree with him, as he knows, but I still think it's great that somebody's so clearly holding the evidential bar very highly. So Brian is a developmental neurobiologist by trade, very highly published with a well-supported lab. His career has been aimed at doing the easy thing of discovering how to build a brain. Easy, not really. But a sort of side gig and a growing interest for him has been, how does the brain generate conscious experiences, including pain? And what does this mean for taxonomics groups like fish, whose brain is very different from our own, both smaller and also embryologically very different in terms of its development. And actually, his paper, boldly titled "Why Fish Do Not Feel Pain," is actually his most highly cited publication. And that's what he's going to talk to us about today. He's going to present the evidence against sentience in fish. So Brian, over to you.
Thanks, Georgia. You mentioned that paper being highly cited. It's quite funny that I've produced some what I think very lovely neuroanatomical work on the development of the brain, but I'm more known for this work, so that's quite interesting. So what I'm going to do is talk to you about pain today, and if you want to escape from that, just take the back door. All right, just follow the fish. So let's see what we're going to do today. All right, so I'll give you an overview of the talk. It's not going to be an extensively long talk because I just want to concentrate on a couple of issues that particularly have been of interest to me recently since that paper back in 2016. So what I'd like to talk about initially is just what is pain. And I think a definition of pain is quite important because it's going to help us understand whether fish have pain or not. And the second part of the talk will be on looking at sort of possible neurobiological mechanisms for pain. Again, if we can sort of tease out some of these, I'm sure that it will help us understand if fish have pain or not. So really just two sections here, very simple. What is pain? And once we know what pain is, what is the mechanism responsible for it within the brain? All right, so let's begin with that first question about what is pain. And the place to start, I suppose, for many people is the field of human pain. And the International Association for the Study of Pain have long had a definition of pain. And more recently, in 2020, they've published a paper that you can see down here at the bottom of the screen, a revised paper where they revised the definition of pain. So I just thought I'd take you through that definition as a starting point for our discussion today. Seem to have a problem with my linkage here. Okay. So their definition, their one-sentence definition, of course, this is a whole paper, but they do have a one-sentence definition here, and it's an unpleasant sensory and emotional experience associated with or resembling that associated with actual or potential tissue damage. That's their one-sentence description, but they've expanded on that description with a number of notes. And so in the paper, they have 6 particular notes that help to describe what they're actually saying here. And the first one is that pain is, they note, is always a personal experience. And hence the word subjective that's often used with respect to pain. And they realize that it's influenced to various degrees by a number of factors, and they can be biological, psychological, and even social factors. They note that pain and nociception are different phenomena. Pain cannot be inferred solely from activity in sensory neurons. They also say that through life's experience, individuals, humans in this case, of course, learn the concept of pain. Now, you have to be careful here what they actually really mean when they say learn the concept of pain. And if you dig down into the paper, what they're actually saying, of course, is humans learn how to use the word from their experience. So from our built experience, people know typically what pain is, and they come to experience pain in very many different circumstances, and that builds up what their definition of pain is through their life experience. And they say that you should respect a person's report. If they're saying they're in pain, you should realize that they are actually in pain. Pain also serves an adaptive role, they note, and it can be have adverse effects on the function of humans and on the function both socially and psychologically for their well-being. So these are some of the notes, of course, I said, that come along with this one sentence description. And finally, verbal description is one of only several behaviours used to express pain. And an inability to communicate does not negate the possibility that a human or a non-human animal experiences pain. So they put number 6 in to open up this idea of pain to animals in general, rather than just concentrate on their typical example being humans. All right, so they concentrate here in this one-liner with unpleasant sensory and emotional experience. So let's just look at that in a little bit more detail. I personally don't like the use of unpleasant sensory and emotional experience. I don't think it's perfect, but that's what their committee has come up with. They chose not to use the word aversive. Initially, there was a lot of discussion in the paper that they should use aversive, but they said that definition was more about the behavior rather than about the experience. And so they really wanted the nature of this internal experience to come across, and hence they used the word sensory and emotional experience rather than keeping in the word aversive. And aversive was initially in their description, and in the paper you follow how this one-liner emerged over time. Now they use the word unpleasant, and they also use the word sensory, but I think the word sensory is redundant here. And I think that pain is an unpleasant experience. You don't need the word sensory in there. I think they put the sensory in there to denote the fact that there are peripheral sensory stimuli that activated circuitry that led to pain. But it misses the point that you can actually have pain without any noxious stimulus at all. And that's called central neuropathic pain. So you can have disturbances of the central circuitry that cause ongoing pain without a sensory stimulus there. Nonetheless, they've kept in sensory in their description. They've also used the word emotional experience. I've always sort of stumbled over this because they actually didn't define what emotional experience was. In their earlier definition, in their earlier paper with their first definition, in their covering notes to that paper, they said that pain was always unpleasant, and therefore, as a result of unpleasant, there was an emotional experience. I'm not quite sure if the two go hand in hand in this case. And if you go to the literature, to try to find out what emotion is, and you'll see it's quite varied what people consider emotion is, particularly what everyday use of emotion is and the scientific use of emotion. And here is a quote coming out of this paper at the bottom here, published just this year in the Annual Review of Neuroscience, a very prominent journal in neuroscience. And they note that among scientists, the definition of emotion remains vigorously debated. So it's a very complicated subjective experience. We consider emotions as, in fact, they consider out of this paper that emotions are functional states. That is, they are not conscious percepts of emotions. They're not feelings of emotions. Because they say that feelings of emotions can only be assessed in humans. And so they refer to emotions really as the behavioral output of central circuitry, rather than the emotional feeling or the so-called subjective experience coming from a sensory stimulus. And you'll see more and more people are actually using this definition to talk about emotions as actually being just the behavioral output rather than any subjective experience. In this context, unpleasant is the feeling and emotion is the behavior. Unfortunately, what happens when you do this, you conflate a phenomenon with its effects. So if you say wind causes tree branches to sway, it says actually nothing about what the wind is. In fact, the wind is the movement of air. So it's like saying pain caused me to be emotional. Right? It doesn't really tell you anything about pain if emotions are in fact just a behavior. So I would prefer to not have the word sensory and really remove the word emotional from this whole description. All right. But of course, many people say, Brian, what you're really actually talking about here is a human pain. You're talking about something that is not really relevant to animals. And of course, that's true. That definition was set up for humans from the International Association, but they did have a caveat for animals in that final note at the bottom. And the common response is that I find is that people say, well, it's not human pain. We're actually talking about fish pain. Fish pain is actually different to human pain. And I come across this many times, and I just thought I'd like to sort of delve into this a little bit better so we can begin to understand what pain is. And this is a classic sort of concept. This is a paper from Lynn Snedden in 2008, where she talks about pain and fear in fish. And she says, we do not argue that fish have an identical emotional experience. And again, this is the word emotional to humans, but we do suggest perhaps that consciousness is on some sort of phylogenetic scale, a sliding scale, so you can move the scale back and forth between animals. And that fish have an experience that is more primitive than humans. It's a primitive form of pain and fear. That same year, Lynn said, however, it's impossible to know exactly what a fish experiences unless one has actually been a fish. And that's a fair comment. And therefore, it is best just to give fish the benefit of the doubt. Now, that's quite an interesting concept in itself, but I just put that up because it contrasts with what she said in the following year. It seems reasonable to view animal pain as a more of a rudimentary experience. So in 2009, she goes back to this original idea that there is a sliding scale, and she thinks that fish, particularly fish pain, is a more rudimentary experience. And again, this idea of phylogenetic sliding scale across species. There was a response by Seith, who's a neuroscientist / philosopher, in a paper, in a brief comment called "Why Fish Pain Cannot Be Ruled Out," and he says that if fish pain exists, it's likely to be qualitatively different and less differentiated than human pain. Again, this concept that fish pain is different, it's primitive, it's rudimentary, it's somewhat different to human pain. Sten then says in 2018, we jump from her work in the 2008 and 2009, and she says, again, pain is unlikely to be an identical experience across species. So it's becoming sort of entrenched into the literature that fish pain is not human pain. Some vets take on the same idea, veterinary's perspective. We could argue that human and animal pain are different, but we should come to some sort of consensus about what pain is, so we can actually still use pain in animals. And so this vet realizes that people are talking about different things when we talk about human and animal pain. Then we come to this really quite insightful statement by a wonderful person who says that peak consciousness is currently impossible to measure and impossible to assess in non-humans. And so, really, where does that actually leave us? Where does that actually leave us when we want to try to define what pain is? So the point I'd like to make here is, if fish pain is different to human pain, and that you can only know what fish pain is if you're a fish, then fish pain can be anything that you want it to be, basically. You can define fish pain how you like, and if you can define fish pain how you like, you can actually say that fish pain can cause all sorts of behaviors, because you can call fish pain whatever you like. You can then say that it causes all sorts of behaviors. And then the reverse is that you can use all sorts of behaviors as evidence of pain. And so I actually don't think that this whole idea of fish pain being different to human pain is actually going to get us very far. And really, I think George has recognized this with this idea that, I should tell you what peak consciousness is. It's a fancy, sexy term for phenomenal consciousness. And phenomenal consciousness is really the sexy name for feelings. And in this case, it's the feeling of pain that is. And so you'll see people say peak consciousness as a shortened form in the scientific literature for pain. All right, so if all sorts of behavior can be used for all sorts of evidence of pain, then really your hypotheses become very hard to falsify in this case. So again, leaving us in some sort of no man's land in this respect. So we delved into this idea a few years back, and we began to address the question of whether we could understand animal consciousness and animal pain. And so we simply defined pain in 2018 in a paper which is put at the bottom of the screen here, Designing Brains for Pain. We simply just said that pain is an unpleasant feeling. And I think that resonates with most people. If you, for your own life experiences, you know that it's a feeling and it's unpleasant. And what really matters, I think, here is that if you're using a sensory stimulus to produce a pain, then it needs to feel like something rather than nothing to be a feeling, right? So this is really important, I think, is that the bottom line here, it's a feeling, and it has to be feeling like something rather than nothing. All right, so if you have no feeling, then you don't have pain in very sort of layman's terms. And we noted in this paper that feelings, of course, share a close relationship with awareness. You have to be aware of those feelings in order to know that you're actually in pain. And so the standard term that most people use these days is that feelings are referred to as a form of subjective awareness. And other people call it an inner awareness. Some people call it a subjective experience. But it's always relating back to feeling like something. And in this case, it's an unpleasant feeling within a subject of interest. Now, this was really driven home in a recent conference that was just held in NIH in the States. And this was a report on that conference, and it was called Next Frontiers in Consciousness Research. And it was a 3-day workshop that was sponsored by NIH, and it brought together a whole bunch of people, experts in very many disciplines, to discuss consciousness. And they really wanted to lay down the current state of knowledge about consciousness and discuss approaches to test some theories that have been proposed for consciousness, and really also to lay out a roadmap for the future of the field. And one of the really important things that this conference realized was that probably more than any other topic, and this is a statement out of the paper, which was reporting on the conference, and it was said that probably more than any other topic in psychology, everyone has their own intuitions about consciousness. After all, it is the most intimate and subjective phenomenon, basically, that we know. Really what is pointed out here is that these folk psychological intuitions, and the word folk often refers to popular. Naive is another word, and it doesn't mean just being simple, it just means it's a popular view. Often with dualistic tendencies, meaning that people actually like to think of the mind and the brain as something different. And this conference really suggested that these intuitions pose significant challenges to rigorous scientific pursuits. The bottom line is this conference came out and said that the core scientific field has soundly sort of come around to understanding that the definition of consciousness it's a subjective awareness. Right? And this idea of a subject being aware of something has guided the field's progress over a number of decades. And luckily, that sort of tied in with our paper in 2018, where we suggested that it should be unpleasant feelings, or in fact, the generic statement of subjective awareness. All right, I'm going to come back. I haven't really defined what subjective awareness is. Of course, I've just changed pain into subjective awareness. And this is one of the problems with the field, actually, is everything's circular. But really, if we let me tease out what I mean by that, and I'm going to shift gears now from laying out this foundation that I wanted to move away from this idea of fish pain being different from human pains. I want to concentrate on the bottom line here. It's a subjective awareness or subjective experience or an inner awareness that's important, and it must feel like something rather than nothing. Right? So we're going to move from that into how can we then begin to understand the mechanism of pain with that basis. And I'm going to take you slowly through some history and where I think we can move on from the current state of affairs. I'm going to take you back very briefly here. I'm not going to do a large history, but I want to go back 400 years. I'm going to go back to a chap, a neuroanatomist, a surgeon, and a philosopher by the name of Thomas Willis, way back about 400 years ago. And many people will remember Thomas Willis if they've done basic anatomy, human anatomy or animal anatomy. There is a circle of blood vessels at the base of the brain that are absolutely important for feeding the brain, and it's become known as the Circle of Willis. So Willis identified this, and it was then tagged in subsequent years as the Circle of Willis. Now, Willis was also really interested in not just neuroanatomy, but he's actually interested in how the mind or consciousness arose out of this massive tissue that he was particularly interested in studying. And of course, in those days, about 400 years ago, this concept of animal spirits was very ripe. And he states in his theses on the particles in the motion of particles in every body, where he discusses this idea of consciousness and where it arises in his discourse. He talks about all bodies being made of a spirit, and of course being made of sulfur, salt, water, and earth. And this was a typical sort of idea at the time. But for consciousness, he was particularly interested in this nature of animal spirits, or some sort of spirit-like nature. And what he defined this as substances that are highly subtle and ethereal in nature. And they were some sort of particle that was present in the body. And hence he called it the infinite motion of particles in every body. And it was this that gave rise to animal spirits and to consciousness. These particles determine the form and figure of everything, and they're prefixed by some sort of divine designation here, which went along with the religious beliefs of the time. So animal spirits for him were present in the brain. When he asked the question, by what workings are they prepared? What channels are they carried at a distance quicker than the blink of an eye? How do they move through the body? And he believed these spirits were passed by the blood into the brain via pores, and through narrow channels they weave their way through the brain to release their energy and become and so consciousness was born within the brain through these spirits or particles, or the motions of particles. Now, some of you may say, well, we've come a heck of a long way in 400 years, whereas others may say, well, perhaps we haven't. And unfortunately, traces of animal spirits still persist in the current literature. And this is a statement from Damasio's work in 2016, referenced at the bottom here. In all likelihood, valence being sort of a positive or negative activity, originates elsewhere prior to the cortex. And we'll come back to this idea shortly. But it's built, this valence is built from a cascade of contributions made by the bloodstream, by critical central nervous system regions devoid of the blood-brain barrier, by peripheral nervous system pathways, and by the subcortical nuclei in the brain, hypothalamus, and basal forebrain. He goes on to say, more recently, minds are made of flowing streams of sensory representations or images constructed with the help of a nervous system, and by the continuous flow of homeostatic feelings within that organism. Feelings emerge from a two-way interaction between the nervous system and non-neural components of the organism. So there's feelings of these spirits that emerge, building from the periphery, passing through the blood into the brain, as building up through the different regions of the central nervous system, particularly at the regions of the peripheral and subcortical nuclei. So this idea of animal spirits is not sort of left us in many ways. Now, others then have taken on this idea that there's these passage of particles, a passage of feelings going through the periphery and into subcortical structures. And so if you have subcortical structures, then you naturally then, of course, feel pain. And K.L. Brown in 2016, who I believe you've been introduced to, said that there is mounting scientific evidence of homologous and analogous structures in the fish central nervous system, including the brain. And so he was referring here to analogous and homologous subcortical structures between fish and mammals. And because there's also this belief that pain emerges from the stimulus and travels through the body. Again, this idea of flowing through the body. And many people, and this is not just K.L. Brown, will typically say that the vast majority of animals learn to associate painful stimuli with specific contexts. And many people will use the word painful stimuli here as if to suggest that the stimulus was generating pain itself, or there was something painful in the stimulus. But in fact, this is just a fallacy. It's a logical fallacy, because the stimulus, to say this, presupposes pain, all right? So you have to say that the stimulus has pain. So the vast majority, what should be said is the vast majority of animals learn to associate noxious stimuli with specific contexts or objects, right? So the stimulus is noxious. And what's the word noxious? It means a stimulus that's going to cause, typically, or has the capacity to cause tissue damage. So it's the stimulus is damaging, but the stimulus is not painful. To call a stimulus painful means that the animal has to feel pain. So it's a logical fallacy in that case. It's also a fallacy to try to prove pain by demonstrating cognitive abilities or intelligence in organisms. And Brown says that animal cognition is a process by which animals acquire, process, store, and act on information gathered from the environment. Nowhere in this definition does it say anything about feelings, and that cognition necessitates feelings or subjective awareness such as pain. To use cognition as proof of pain requires two starting premises. One, that fish feel pain. So if you're going to use cognition as a mechanism for proving pain, you have to say that fish actually feel pain to start with, and you actually also have to say that pain is responsible for cognition. Right? So two premises that require starting point to say that intelligence or cognition is a demonstration of subjective experience. It's also a logical flaw to suggest that because pain can perform certain behaviors that seem dependent on pain in humans, that fish then should feel pain. So let's sort of tease out this a little bit more. Let's take if A causes B in human, and A being pain, and if pain causes behavior in humans, B is observed in fish. It doesn't follow that A causes B in fish. That's just a logical fallacy to suggest it does. Let's look at it in a simple analogy. Batteries A cause movement B in Tesla cars. Movement B is observed in Ford cars C. It doesn't follow that batteries cause movement in Ford cars. So it's a fallacy to suggest that that otherwise. So interestingly, some of the same people who follow this logic also say that fish pain is different to human pain. And because fish pain is different to human pains, then you can make this leap of faith. But in fact, if you say that human pain causes B behavior in humans, B is observed in fish, it doesn't follow that fish pain causes B in fish. It's another logical fallacy. So one has to be careful when we make these leaps in trying to identify pain in fish. So I want to move away from these folk intuitions, and that's what that idea of this naive or folk intuitions that the conference was trying to move away from, and really look at what's modern neuroscience is actually telling us. And I think it's really providing some wonderful insights into the circuitry underlying subjective experience or subjective awareness. So I'd like to take you through a little bit of history first, and then move on to some more sort of modern approaches. All right, this is a paper that is a review paper, just came out in Nature Reviews Neuroscience, a very high impact journal. And I think it really sets the scene for where neuroscience is with respect to pain. The title is Neurocortical, or what we're talking about really here is cortical circuits in pain and pain relief. And neocortex really referring in many respects to mammals. And if you read this abstract, and I'll just read a few things about this abstract, and it says, for instance, emerging insights into neocortical pain processing of pain in the cortex suggests the existence of neurocortical causality and specificity for pain in the level of subdomain circuits and cellular entities and the activity patterns they encode. I won't go into the details of this abstract, but the bottom line, this paper is defending the idea that modern neuroscience has moved on from subcortical regions to the cortical regions in vertebrates for the analysis of the circuitry underlying pain. And I'll give you some examples of that shortly. Oh, I'll give it to you right now, in actual fact. And I've just pinched a few papers out of the recent literature that really highlight some of the advances that are being made. And these typically, these insights are coming from high-profile papers, journals. They're not coming from very obscure journals. These are really leading papers in the field. And this paper in Science Advances in 21 was looking at pain and looking at the circuitries underlying pain in the somatosensory cortex. And some of the techniques are quite wonderful. And we've got these wonderful techniques of activating specific circuits using transgenic approaches with chemogenic activation of these circuits. So you can actually use drugs to activate circuits that have been modified transgenically to become active in the presence of certain drugs. And using these drugs, or deactivated in the presence of certain drugs, it's been shown that the somatosensory cortex is important for pain and we're talking about mice here. So all this work, of course, is in mice. The transgenic approaches are being used in mice, but it won't be long before we see the same transgenic approaches applied in primates. And there's a lot of interesting potential work coming out of China at the moment in this area. This is a paper in interdisciplinary science, eye science, and again, using these chemogenetic activity and inhibition of somatosensory. So it's backing up a separate group, backing up this work of the role of somatosensory cortex in pain, the circuitry involved here in the generation and modification of pain. Cell reports last year, again, they used a chemogenic activation, and they didn't actually attack the somatosensory cortex in this region. They created transgenic animals in which they modified the dorsomedial prefrontal cortex, and in particular 3 regions, the rostral anterior cingulate cortex, come back to that shortly, prelimbic cortex, and the insular cortex. So all cortical regions they could activate and deactivate using these chemogenic approaches, and shown to be involved in pain. Some really wonderful work occurring in this area. And this, again, a paper in Neurone, one of the high impact journals in the field. Again, they looked at the dorsomedial prefrontal cortex, a different group, and they've shown using similar techniques that it contributes to pain as well. So this is the level of detail, and I'm not going to go into the details here because it's pointless, because it would take far too much time. But the bottom line here is you can activate specific pathways in the cortex to produce or modify pain in mice. So what have we learned from humans? Of course, we can't do this sort of work in humans, and we have to rely, of course, in many cases on neurological cases or stroke cases. And there's a whole vast literature on this area. And I'm just taking one here because it's quite a lovely image. This is a slice through the brain of a human. And what you see, this white area here, is a stroke in the left insular cortex, a beautiful, very specific localized stroke in that, which really means the area is dead. It fails to, the blood vessels there have burst, and the stroke there has caused death of the insular cortex. And what this patient then was unable to feel cold pain or pinprick pain on the contralateral side of the body. So it's a very nice demonstration that if you manipulate the insular cortex, which is one of the regions that those previous papers were looking at from the dorsomedial prefrontal cortex and the insular cortex, that if you manipulate that area in an animal, you modify pain. If you manipulate that area in a human, you modify pain as well. You lose pain in this case. And there's a whole bunch of techniques that are used to address this in patients now, and in particular patients who have serious pain produced typically in cancer, the end stages of cancer. And so there's a real desire to actually alleviate these patients from severe pain without the use of drugs. And one of the approaches here is where you lesion the spinal pathways that deliver noxious information from the periphery to the brain. And so this purple area here is the anterior lateral quadrant of the spinal cord that has been lesioned with electrical stimulation. So you come in with a needle, electric probe, and you blast this anterolateral region of the cervical spinal cord specifically in an attempt to ablate the pathways carrying information from the spinal cord to the brain that leads to pain in the brain. And another approach in this same paper was to lesion four very precise places in the anterior cingulate cortex. So this is the anterior cingulate cortex and the prefrontal cortex in humans, 4 lesion sites here. And what they found is before the lesion site, patients with this lesion, before the lesion site, they had incredible pain rating, the highest pain rating of 10. And after that, it went down to 0 with lesioning 1 month later after lesioning of this pathway. If you ablate just 4 regions here, 4 very precise regions, you can dramatically reduce the pain down to a level of around 2 from 10 in these patients. It's not completely ablated because that's just one region in the brain that's involved with pain. Whereas this region here, of course, is delivering the information to the brain from the periphery. That work was published in Brain just this year. And this is a typical circuitry that we see. This is a circuitry defined by Coghill in 2020. It was published in Trends in Neuroscience. And you've got, really, I'm not going to go through all this in detail. I just wanted to show you that there's a network of cortical regions, including somatosensory areas, the insula again, and the anterior cingulate cortex, and a subcortical region here, the amygdala. Here's the spinal cord. Here's the anterior lateral spinal quadrant. Quadrant is delivering the information to the thalamus or to the brainstem, and from the thalamus it's going up to the cortex, and the brainstem is going to the amygdala, and from the amygdala across into this network in the cortex. Now, of course, if you ablate the amygdala in humans, and there are cases of very complete bilateral ablations in humans, pain remains. So you can't get rid of pain in these patients. And so this is the pathway going from the spinothalamic pathway through the thalamus into the cortex. And as we've shown before, if you ablate particular regions of the brain, you remove pain in humans. So what I'm typically faced with when I present this work, and I'm sure most of you are thinking the same way, or many of you are thinking of the same thing, that fish feel pain in other regions, right? So again, fish pain is different to human pain, so therefore fish pain must be in a different region than human pain, and that's the logic that's driving that line of thought there. And so Damasio says, based on anatomical and physiological evidence, subcortical structures, even the peripheral and enteric nervous systems, appear to make important contributions to the experience of feelings. Enteric nervous system is a part of the peripheral nervous system, but to him, it appears to make important contributions. And of course, the important contributions are delivery of information from the periphery, of noxious information from the periphery to the brain. And then Devor says that the possibility of subcortical activity being the primary substrate of pain ought to be taken seriously. Again, we're just making guesses here. Appear to possibility. Merkus says that the tectum and the periaqueductal complex may have information capacity and connectivity, requiring a candidate, in this case fish, to have elementary, again, these sort of strange rudimentary feeling states. So there's a large belief out there that it's the subcortical regions in fish, and even if we can go and prove that humans have cortical pain, but fish feel it in different brain regions. So let's look at different brain regions to try to find out where this is happening. And this is a typical early model for fish, where a lot of ablation work is being done. So like we have stroke in humans, or we have electrical ablations of regions in cancer patients. Similar experiments can be done in goldfish. And this is the front end of the brain, the olfactory bulb, the telencephalon here, or the pallium, and we've got this large structure here is the optic tectum. We've got the cerebellum, and they have a very large, goldfish, as opposed to some other fish, have a very large vagal lobe here, determinations of cranial nerves in that lobe. And then you have the spinal cord, and you've got the hypothalamus here. Now, so what I've just done now is people have undertaken classical lesion studies in goldfish, and let's just cut out this whole area here and ask what happens when you do that with animals. And here are a couple of papers here that looked at doing serial ablations, and they've ablated the telencephalon. And so here's what an ablated telencephalon looks like. Here's this goldfish here. And that's the telencephalon. These are the optic lobes here. That's the cerebellum, and that's the vagal lobes here. And these out here are the olfactory portions of the animal. Now, if you take out all this, you've ablated that, you're left with this, really the remaining optic lobes, vagal lobe, cerebellum and vagal lobes. And you avoid active learning if you look at active avoidance learning in these animals. They lose it after ablation of telencephalon, but they don't lose the response to electric shock. Right? So they lose the learning, but they don't lose the ability to respond to the shock. Right? And so animals can actually be retrained to perform active avoidance in the absence of the telencephalon. Right, so the response to the electric shock remains. So it's not. So if animals were feeling pain, it isn't happening in the telencephalon. Let's take out the tectum now. So we've taken out this large tectum. It receives predominantly information from the eyes. If you take out that, you do a bilateral tectal ablation. Here's the papers that have done that below. I should say basically they still condition to electric shock. So you can still condition animals to electric shock in the absence of the tectum. So it's not tectum. If pain was responsible for the conditioning, it isn't happening in the tectum. It's not happening in the telencephalon. So where else? So let's get rid of the cerebellum. So let's cut off the cerebellum. Here's some papers that cut off the cerebellum. Cut off the cerebellum. We know that cerebellum is responsible for the eye blink conditioning using electric shock. So you can condition animals to respond to a light if you give them shocks coordinated with that light. And if you remove the cerebellum, they no longer can be able to perform that conditioning, but they still respond with eye blinks to the electric shock. So it's not pain in the cerebellum that's driving that. There's some other papers here that actually did quite sophisticated ablation of granule cells and Purkinje cells within the cerebellum rather than the whole cerebellum, and they got the same results in zebrafish. So what are we left with? What are we left with is that we've got the hypothalamus that is predominantly left, and we've got two large areas here in the brainstem, the pons and the medulla, that could be driving pain if pain was being experienced in these animals. And I've just looked at my watch and I notice we're running out of time, so I'm going to have to move a little bit faster here. Bottom line is the hypothalamus consists of a number of small nuclei, and I've given you the names of the nuclei here. They don't really concern us very much, but except we're going to talk a little bit briefly about this nucleus here called the preoptic nucleus of the hypothalamus, just sitting in front of these tiny sets of nuclei here. That preoptic nucleus has been shown to drive locomotion, and it drives locomotion by direct connections with hindbrain premotor neurons. So in that pons hindbrain region that I showed you before, there are sets of premotor neurons that then drive spinal cord circuit generators to produce locomotion. So it's a very nice projection from the preoptic nucleus, and that's been worked out in animals using ablation, chemical ablation in specific populations of neurons to work out the circuitry driving the locomotive behavior in response to a nociceptive behavior. And what they've actually been able to show very recently in zebrafish as the model, because these fish are very easily used as transgenic models for understanding the circuitry, that there are a population of 70 neurons expressing a neuropeptide called oxytocin in the preoptic nucleus area. And that area, these neurons are responsible for that circuitry for the nociceptive heat activating locomotion and removing of the animal away from the source. So stimuli coming from the spinal cord after heat applied to the animal activates this population of oxytocin neurons, and they connect directly with the hindbrain premotor neurons, and those premotor neurons can be optogenetically activated. So you can actually activate these neurons with 25 of these 70 neurons you can activate with laser light to drive this circuitry. So only 25 of the 70 neurons are needed for that animal to move themselves away from an nociceptive stimulus. And these premotor neurons then project to the spinal cord. You can actually ablate this whole region of the brain, which they've done, and just activate these premotor neurons with oxytocin directly. You can sprinkle oxytocin onto these neurons, and you'll get the locomotor reflex generated. So it's a very lovely reflex that's driving this. And that's, for some reason, that won't come up. Let's go back. Bottom line is another paper just published a couple of years ago has been looking at this in more detail. And again, I'm not going to go through the transgenics, but they've identified two populations of neurons. One of these are the oxytocin. This is a different group, and another small population of neuropeptide expressing neurons called CRF. And they're labeled here in purple, so you can actually see them in the hypothalamic regions. If you activate these neurons chemically or by laser, you can produce a locomotive behavior. So again, this is demonstration that these neurons in the hypothalamus are generating the behavior. So they're sufficient for the behavior. If you ablate these neurons and apply heat, the animals no longer respond. So it's necessary. So this pathway is both necessary and sufficient for producing the locomotor response to a nociceptive behavior. This is all subcortical, right? You can remove the rest of the brain and it makes no difference. And of course, they've gone on to map the circuitry. So there's some really lovely projections of these neurons. And then now these purple neurons are the neurons in the premotor area of the hindbrain that then drive spinal reflexes in the spinal cord. So let's do a quick summary. So we're going to start with pain in mammals is generated by a neural network in the cortex. And that involves at least this circuitry, two-way circuitry between the somatosensory areas, anterior insular, posterior insular, and the anterior cingulate cortex. There are other areas, but they're the principal areas driving that circuitry. And that's now becoming well mapped out in mammals. In fish, classical lesion studies have localized nociceptive processing to the hypothalamus and hindbrain. That's the classical lesion studies. And then we've followed that on with molecular genetic studies, and we've identified a pathway that goes from the nociceptive forward to the preoptic, go to the premotor area to the spinal cord. That's a feed-forward pathway, and it doesn't rely on feedback. And it's these feed-forward pathways are present throughout many areas of the brain and areas of the brain that are not conscious, even in humans. So this is not good evidence for pain in fish. All right, is Georgia still with us? Georgia, are you still with us?
Yes, absolutely.
I've actually gone way over time, so I can stop right now because I could keep talking, and I'm not sure if your seminar should be finished in 50 minutes or what, 55 minutes?
Well,
what's your
the slot is kind of officially an hour. So what I was going to do is say at 5:30, perhaps tell people about the next seminar, remind people this is online, and then we can chat and people can stay on.
Okay. Well, why not look, if I start now, I'll just keep going, right? So what I'll do is just sort of, if you give me 2 minutes, I'll just quickly sort of show people where we're going. Right.
Yep. Cool.
Okay. So just give me 2 minutes, and we'll quickly. And I apologise to the audience for keeping them here. I know they want to go home and make dinner, so let's just quickly go through an idea here. So really, the idea that we're working on now is that if we could pinpoint the circuitry that underlies subjective awareness, that subjective experience in the cortex. So I think maybe we can then see whether other animals have a similar circuitry that could be driving that sensation or subjective awareness. And I don't care if it's in the cortex or not, but we can only do it in mammals because we know where the circuitry is in mice and in humans, and then take it from there to other animals. So that's really the approach that we're adopting at the moment. And I'm going to skip through all this stuff, which is, and say that we're working on this idea that that you have an input, some processing, and an output. And this is electric shock to the hypothalamus, to the hindbrain in animals such as fish, that is both necessary and sufficient for behavioral outputs. We're suggesting that somehow there's got to be some other circuitry lying above that that acts as the starting point for subjective awareness. And if people are interested, I can talk about it later, but that circuitry involves predictive models that then have very complicated circuitry that is not existent in fish that could very well produce the sensations of subjective awareness in humans. And so I want to leave you with this idea that if we can understand awareness, the ability to be aware of what we're processing. So here we have the processing pathway, but if we become aware of that, then we may be able to understand what sorts of animals have the capabilities of subjective awareness. So I'm just going to leave it like that and show you that we've got some of the circuitry in humans that matches very much with that. And so the take-home message I want to leave you with is we shouldn't conflate nociception and pain. It doesn't help us. It doesn't help us to say fish pain is different to human pain. It really takes us nowhere. Folk intuitions about conflating cognition with pain doesn't help either. And what we're really stressing here is if we can begin to understand the structure, and the structure is so important for function, it's going to help us to understand which animals are capable of subjective experiences. That's really where I want to. Thank you very much, Georgia.